Здравствуйте, уважаемые коллеги. Павел Михайлович немножечко приболел, поэтому попросил меня провести это. Что у нас, заседание? Нет, не заседание. Ну, лекцию же не я буду вести, семинар. Да, давайте, я буду вести этот семинар. Вот, и it's my pleasure to introduce good friend and long-time colleague, Professor Jim Wilson of the University of Edinburgh. Should probably tell a few words about you, Jim. Good words, obviously. And well, a few things to mention. I think, Jim, your career is centered around human population genetics and human genetics of quantitative traits and complex traits, right? And one of the things, well, probably there are two things which are visible if I look into your papers, right? So I see your name a lot on different genome-wide association scan papers, and that's obviously linked to the ORCADES and now ICAN cohort, which you are principal investigator and, well, chief, driving force behind, right? And then another thing which is quite visible is your interest in population genetics and specifically in inbreeding and effects of inbreeding onto complex traits, right? And well, doing work in this area, Jim been very successful. So he is one of these highly cited scientists now for 5 years, probably, right? And with very nice Hirsch index of 80 something. Yes, so that's nice, right? And well, going back to science, you're going to talk about inbreeding and human complex traits, which is obvious from the title. So, Professor Wilson, the floor is yours.
Thank you very much, Yuri, and thank you very much, everybody, for coming. I've been to Russia twice before. This is my third visit, and I'm always very delighted to come here. I've never been to Akademgorodok or Siberia before, but I love it, actually. It's amazing place, a well-kept secret, I think. Anyhow, it's quite different from where I grew up in Scotland. Scotland's famous for rain and wind and whiskey, and yeah, it's nice. I come from considerably far north of here. I grew up at the 59th parallel on a small island, and maybe that explains my interest in some of the topics I'm going to explain to you today. So I've entitled my talk Big and Clever, Inbreeding, Fertility and Health. So if we think about it, I don't suppose anyone in this audience would be particularly surprised to realize that we are all related. People in the general public have this idea in their mind that we're all related. In Scotland, we have a phrase, we say, we're all Jock Tamson's bairns. So bairns are children, and Jock Tamson's some sort of mythical ancestor that the people of Scotland had. But what does this mean? What does it actually mean to be related when you think about it? Of course, it means that we share common ancestors between the two people who are related. So if we just take some random person, like the boss here, born in the 50s, and think about his ancestry, like all of us, he has 2 parents, 4 grandparents, and 8, 16, 32, and so on and so forth. And very, very quickly, as you can see here, you get to be hundreds of people, thousands of people as you go further back in time. And it's this huge increase that reaches millions. So by 20 generations ago, so circa 1330, Vladimir has 2 million ancestors. So this is at the time, around about the time of the Black Death, was sweeping through Europe. And the population of what's now Russia was actually less than 2 million at this point in the past. This is called the ancestor paradox. And the resolution of this paradox, as you possibly are aware, is obviously that some of these people are the same people. There are not enough people in Russia to be the ancestors that are required, so some of them are the same people. And this is why everyone truly is related, and everyone's parents are related as well. Mine, yours, Vladimir's, all of ours. So that's the case I've made for Russia. I could make the same case for Scotland. But what about all of the world? What about someone who has slightly more diverse ancestry than perhaps I do, or perhaps Vladimir does? What about Barack Obama? So his father is a Luo from Kenya, and his mother has actually Scottish, Irish, Welsh, and English ancestry. So he's a good mix, I would say. But because of this exponential rise in the number of ancestors, it's simply a matter of going back a few more generations. By my calculations, if you go 29 generations ago, you should have 500 million ancestors. So we're back now to about the year 1090. So in England, William the Conqueror had just conquered England. Over here, we're 2 generations after Yaroslav the Wise, the Grand Prince of the Kievan Rus, just before or around the time of the First Crusade. So that's as far back as we have to go before anyone in the whole world is in theory related to one another, because the world population at this time was less than 500 million, was about 300 million. And what's also true is because of this overlap in ancestors about 1,000 years ago, anyone from that time who is someone's ancestor today is actually everyone's ancestor. Many, many people from that time have no descendants, but those who do are actually all of our ancestors. So it means it's true that we all descend from Charlemagne, we all descend from the King of Scotland, Kenneth MacAlpin, we descend from Alfred the Great of England, indeed we descend from Yaroslav the Wise, and many, many other people, famous or not famous, as long as they have some descendants at that remove of time. And some people say to me, I don't believe you, Jim. I don't think that's true. All my ancestors come from one place. I'm not descended from Charlemagne. I know all my ancestors come from wherever, fill in the place. And the reason why they're wrong, and I'm right, I hope I can explain to you here, showing this simulation that Graham Coop carried out. We've got 10 populations here. I've decided to use the city of Tomsk as the example rather than Novosibirsk, because it has a slightly older history. And we're simply simulating where people's parents are from. So you've got a 1 in 50 chance that your parents come from the next deme, from the next town, and a 1 in 5,000 chance that your parents come from far away, from the 10th one, for instance. Yet, this means that something over about 98% of the ancestors are local. And so what happens is that obviously you've got 2 parents, 4 grandparents, 8 and so on. They're mostly coming from the same place. This person's ancestors are all from Tomsk. But if we go back a little bit further, 1 great-great-grandparent is from Moscow. And so as we go further back in time here, obviously all of the people in Tomsk deep in the past eventually are taken up and become this guy's ancestors. And then the same thing happens with Moscow. You get a little bit further back, 9 generations ago, beyond this person's knowledge, someone had moved from London to Moscow. And you start filling up all the possible ancestors from London. And you keep going back, someone had actually moved from Africa to London, and so on and so forth. And you start, even with very little migration, to start covering the world in terms of who is your ancestor. The specifics of the time back obviously depend on the exact migration rate and the exact population sizes. But because you're doubling each generation, after a while, it doesn't matter. There's simply so many that this becomes true, whether you believe it or not. And part of the reason for this is because in the past, people took wives very close at hand. These are real data here. They're actually from Orkney, the island I grew up on. But this would be the case in many, many places prior to the age of urbanization. What someone did was to look through all the marriage certificates for 100 years and map out how far apart the husband and wife were born. And the mean distance is about 1.5 miles. Very, very close at hand. My colleagues in Finland have much nicer data than this, much larger data sets, so they can slice it up through time. And they saw exactly the same pattern as I did until about 1889, and it suddenly jumps. The distance jumps up to 9 to 10 miles. And it's because the bicycle had arrived in Finland, and men could go that little bit further to find a wife. So I was going to talk to you particularly about inbreeding. I hope I've begun to show you that we're all inbred to some degree, but in many cultures and places, people actually marry their very close relatives. Here's a famous fellow born in this sort of mid-century era, and you might know that Prince Philip and Queen Elizabeth are second cousins once removed. They're also actually third cousins, and they're actually fourth and fifth cousins in very many different ways as well. Prince Charles only has 28 instead of 32 great-great-great-grandparents. And this is the beginning of something that we call pedigree collapse. Rather than having all of these ancestors that you expect to have, your pedigree collapses back down again into a smaller number of ancestors. And it's not just Prince Charles and the nobility who practice consanguinity. This is a pedigree of a volunteer in one of my studies. And you'll see that this is actually a very unusual pedigree if you're used to looking at these things. Here's my volunteer. He's got 2 parents, obviously. He has 4 grandparents. Indeed, he has 8 great-grandparents. But if you go further back in time, there are some very unusual things that you don't see very often, wherein, for instance, here, he descends from 4 siblings in one family. Here again, 4 siblings in one family. Here again, 4 siblings in one family. And here again, 4 siblings in one family. So it's quite an incredible pedigree. So despite this volunteer's parents' closest relationship being third cousins, he actually descends 18 different ways from Andrew here. And see Andrew up at the top. Andrew was only born in 1708. It's not that long ago. And the pedigree collapse here is incredible. Rather than 64 great-great-great-great-grandparents, this fella only has 20 of them. So it's gone up and then really down like this. And people, as I said, ask me why I'm interested in these things. I think probably the key reason is because this volunteer is my own father. And the reason his pedigree looks like this is because he was born on this rock in the ocean. It's 39 kilometers from the nearest land, and it's 1 mile wide and 3 miles long, and about 50 people live on it. So this is why I'm quite interested in consanguinity. And it's not just me, because others around the world do surveys of this kind. Alan Bittles in Australia carried this out, looking to map across the world where people practice inbreeding. And it won't surprise you to see that this is concentrated in the mostly Muslim lands of the Near East, the Middle East, and the Maghreb, where in some places more than 50% of marriages are contracted between first or second cousins. But in other places as well, here and around, there is a certain level of inbreeding in the population. So Charles Darwin, actually, was one of the first people to be quite interested in this. And Darwin said, nature tells us that marriage between near relatives is in some way injurious. And like me, Darwin had a reason to be interested because, unlike me, Darwin married his own first cousin. Emma Wedgwood, as you can see here, shared a pair of grandparents with Charles. And they had 10 children, some of whom were quite ill. And Charles spent quite a lot of his time worrying about whether this fact that he'd married his cousin was to blame for some of the diseases that his children had. Others have studied this as well with greater and lesser degrees of success. Raymond Pearl said in 1920 that no phase of biology has been enveloped in such a fog of superstition, old wives' tales, and other sorts of misapprehension as has inbreeding. So I hope I can clear some of this superstitious fog for you today. As I said, many scientists have studied inbreeding, in the traditional method of comparing the offspring of cousins with the offspring of unrelated parents. And of course, this leads to an increase in the rates of various rare recessive conditions, diseases like Tay-Sachs disease, principally among Ashkenazi Jews, phenylketonuria, albinism, cystic fibrosis is very, very common in Scotland and Ireland. There are lots of birth defects. And it's been well known, these things, for a long time. What's unclear is whether there's actually an effect on the more common diseases, the diseases that most of us in this room will fall foul of as we age. And that's where my interest lies. I also have approached this in a rather new way. Rather than using pedigrees and comparing sets of people who are inbred with those who are not, we want to do it genomically using DNA. And this is because family trees, they're often wrong. Some people don't know who their father is, and they're usually rather short. You can only go back so far in time before you run out of records. And as we see, because lots of these connections are deeper in the past, it's important to know all of that. So I spent about 10 years looking at this using DNA methods, and you don't need a family tree then. We could apply our methods even to Neanderthal DNA, mountain gorillas, you name it, anything for which we have a reasonable genomic readout of. So let's go back again to the idea of relationship, kinship, and what it actually means. As I said, it means you share an ancestor, but it also means that you share DNA. So we have a couple of printouts here. This is the sharing on the left-hand side in blue between me and my uncle. So we share about 24% of our DNA in various chunks across these stylized representations of the human chromosomes. If we look now to the middle, this is me against my first cousin once removed, so quite a step down in kinship, and you see there's a step down concomitantly in the amount of DNA that we share. Fewer blocks, shorter blocks, 4% in total. Come now round to the right-hand side. This is me and my fourth cousin, so we're getting quite a lot further out here. Only one block shared, 0.1% of our DNA. But remember, the genome is large, very, very large, 3,000 million letters of DNA. So it's still 9 million letters that I share with my fourth cousin. So where do these come from? They come, as I say, from our common ancestors. So there's a very simple pedigree here that you might or might not be familiar with. Here we have the offspring of first cousins. And as you can see, he has two common ancestors here, his great-grandparents. And so when this sort of mating occurs, when these two, who are first cousins, marry one another, sometimes they then pass down these different chromosomal chunks that I've colored in red and in blue here, down the generations like this, such that the same piece of DNA is inherited down both sides of the family, and this then generates a segment of DNA that's identical in the offspring of cousins. So this is exactly the same pedigree as Darwin's children, for instance, would have. And you can see here is a piece of DNA that they've inherited identically from their great-grandmother. Here's a piece of DNA inherited identically from the great-grandfather. So these segments we can read out very simply in genomic DNA. And you see them here as a run of consecutive DNA letters where both copies are identical. So if this is a piece of DNA that I have read from an individual, we've got two copies here, one from mum, one from dad, and they're different, usually different. We've looked at these bits of DNA that vary and they're a little bit different. But here, suddenly you have the onset of a run, a string of letters that are exactly the same on the maternal DNA and on the paternal DNA. And then against different again, and here again we have a run. And this state of being identical in both copies of your DNA is called homozygosity. It's a bit of an ugly word, it's a Greek word, but we use it. And we call these long segments runs of homozygosity because they're simply that. They're a long run of homozygous DNA letters. And these letters are the ones that have been inherited from the common ancestors. And they could be long, as I've shown here, or short. And this is because they could come from more recently in your pedigree or from longer ago in your pedigree. The longer ones are more recent, the shorter ones are from longer ago. So why do we study these? I've already alluded to the fact that there are certain kinds of genetic variant, as you probably know, which only have an effect when they're present in two copies, and they don't have an effect when they're present in one copy. This is obviously how traits skip generations, like red hair or cystic fibrosis and these things. And clearly the more ROH we have, the more of these runs of homozygosity, the more likely these kind of variants are to express their effects. We've already talked about some of the diseases, the genetic diseases, the Mendelian diseases, the simple diseases that these variants can cause. But I'm interested, as I said, in whether they also cause complex diseases. Do they increase the risk of heart disease, cancer, diabetes, stroke, osteoporosis, glaucoma, these sorts of diseases, particularly given that about 10% of the global population, nearly 1 billion people, practice consanguinity. They choose to, or their culture chooses for them, that they marry their cousins? I think this is an important question, and it's a question that no one really has been looking at. These ROH also, by the by, provide information on the population history, on the demographic history of the population. Now, Yuri has already mentioned a study of mine called the Orcade study, based on these islands here, where I grew up, in the north of Scotland. And as part of this study, we were looking at ROH. So we took participants from these islands with a 6th-generation pedigree, so quite a good pedigree. We knew who all their ancestors were, and we divided them up into different categories. We had the people who were the offspring of first cousins and second cousins. Then we had the people who were not the offspring of cousins, but all of their grandparents came from one place. For instance, if all 4 of your grandparents came from the Isle of Westray here, or if all 4 of your grandparents came from the Isle of Sandy, we would call these people endogamous. So we have the inbred people, A. B, we have the endogamous people. And then we had another group where all the ancestors came from Orkney, but maybe one from here, one from here, one from here. They were from different parts of Orkney. And we had a final set of people who had one parent from Orkney and another parent from the mainland of Scotland. So these people, we were able to check their pedigrees and see they did not have any links between the maternal and paternal side, and they probably didn't have links going back at least 10, if not 20 generations. So this was our test set, and we used these to look for the first time at runs of homozygosity. And what did we find? This is a very simple diagram of chromosome 1, so the longest of our human chromosomes, and we're comparing here the set of half Orcadians, so these are the people who have got one parent from Orkney, one parent from elsewhere, to a set here of the offspring of cousins. And you can see immediately that the offspring of cousins have more of these ROH. These ROH are longer, and they're more random, compared to the people who we are demonstrably are outbred, people who have no shared ancestry for 10 or 20 generations. They have very few ROH, they're very small, and they have a slight tendency to be in the same place. So they're very different across our genome. And what we realized is you could add up the lengths of these ROH here and derive a measure of how closely related the parents were, an inbreeding coefficient, which we called SROH, the sum of ROH. So when we then look at the sum of ROH along the bottom here of the graph versus the number of ROH. So remember that each person has quite a few of these of varying lengths. You add them up to get the sum. Obviously, there's a correlation here. But what we were very pleased to see is that there was a very nice distribution in that those in blue here are the people who are the offspring of cousins. Those in red are those who are endogamous, where all 4 grandparents come from one small island. Those in blue, where you're more random Orcadians, people who've got maybe a granny from here, a granddad from here, and so on and so forth. And those in black were the half Orcadians, the people who were demonstrably outbred. So there's actually a very close relationship, both between the inferred demographic history and also with the pedigree. We could, as I said, measure the pedigree inbreeding of these people, and there was a correlation of 0.86 in Ruth McQuillan's work here from 2008. So we then decided to look beyond the shores of Orkney and take our method out to the rest of the world. And we were able to use the Human Genome Diversity Panel, which is a selection of DNAs from 52 populations around the world for 1,000 people in total. And we're plotting the exact same plot here, where we summed up the total length that is identical, the total length in runs of homozygosity versus the number. And what we could see was that the continents differed from one another in quite striking ways. I've only plotted a few populations here to make the graph simpler to understand, but these populations represent their continents. And the most striking thing of all was that the South American tribal populations, exemplified here by the Caritiana, who are from Brazil, from the Amazon jungle, were incredibly homozygous, to the point that it was worrying to me that these people were wandering around. This person here, I think these 3, can you see? You're not, anyway. These 3 guys out on the extreme right of the graph are probably the offspring of an avuncular relationship, where an uncle has mated with his niece, and one of them has the entire chromosome 1 from end to end entirely homozygous, which would surprise many clinically trained people that this would be a viable human being, let alone entering a study and perhaps giving consent. And the Caritiana are really the extreme of any society that I have looked at, but all of the Native American populations were very, very high. Here, I'm imagining that you're seeing this red thing, but you're not, anyhow. So if we move in from the Caritiana into the green, here, it just takes a long time to catch up, into the Papuan population here, from the Papua New Guinean highlands, you can see that they have a very large number of ROH, but they're not quite as long, each one, as that in the Caritiana. So it pulls them more over to the left. And I think this is because of a history of very small population size in that part of the world that's generating lots of ROH that are from long ago. The shared ancestry is very deep in the past, but there's rather a lot of it. We then move down to find a set of East Asian populations, Japanese and so on. Below that, a set of European populations, French and so on. Below that, then in green, a very interesting population, the Baluch from the tribal regions of Pakistan. And you see there's a very high variance. Each circle is one individual human being. And you can see that the Baluch go all the way across the spectrum from being really very outbred, if they happen not to be the offspring of cousins. And there's a very strong right shift where you're pulling the point to the right on the graph, which is a classic sign of inbreeding, because cousin marriage creates a small number of very long ROH. So it pulls individuals to the right across this graph. And we know that people in this part of the world have a tradition of marrying their cousins. Down at the bottom, near to the origin of the graph, were most of the African populations, exemplified here by the Mandinkalou from Senegal. Then I genotyped another guy, and he had no ROH at all. Can anyone guess who he was? He was a guy I was involved in a TV program with, and his father was Irish, and his mother was Yoruba from Nigeria. So he's a first-generation F1 hybrid mixed race individual, and he literally had no ROH because his mother and father don't share ancestors until very, very far back in time. We did him on a better chip, and we did eventually find a few small ROH. This is another global survey from a review paper that we recently wrote, and I just wanted to highlight it here. It shows the same patterns where we have the Caritiana, look right up there at the top right, but also other Native American populations. But perhaps of interest to this audience are that in purple here, it's working, 17, 18, and 19 are really quite high homozygosity populations, the Koryak, the Selkup, and the Chuchi, who obviously all come from this part of the world. So it's not only Native Americans, there are other tribal populations who have this pattern in their genomes, but by no means all such populations. It's very specific to certain groups who obviously have a tradition of consanguinity or a very small population size. The guy that we found with very few ROH wasn't unique. I became interested in this and recruited various friends, and I put a couple of famous people in there who are also F1 hybrids to look at this. And the pattern held up very much as we expected, such that the genetic distance between the parents was quite predictive of how few or how many ROH someone would have. So you can see here, remember, the Europeans were quite low on the world scale, but all of these various kinds of mixed race individuals are much, much lower, with West African crossed with non-African being the very lowest of all, just like the first guy that I ran into. So we can learn a little bit about population history from ROH, but we're more interested to see whether it has any effect on the phenotype. Does it influence the complex traits that we study every day? So like most people, we started with height. It's not the most interesting trait in the world, but it's a very classic trait being studied by Galton, by Fisher, and it's very, very repeatable. It's easy to measure height, unlike measuring blood pressure, and it's very commonly measured. Everyone measures height, and it's also a good trait because it's very genetic. It's well known to be about 80% genetic. So Ruth McQuillan, my former PhD student, decided to look at this, and we regressed height on the sum of ROH while controlling for age and sex and other such factors in the ORCADE study and in a number of other studies, 20 other such population-based cohort studies. And we were delighted to find that the more ROH people had, the shorter they were, with a very nice p-value that we published as the first foray into this kind of work, trying to use genomic methods to understand the effect of ROH on complex traits. We then moved forward after a couple of years of sorting out the methodology so that we could apply it to different kinds of chips and different kinds of data, and created a consortium which we call RhoGen. And in its first iteration, around about 2015, we decided to look at 16 traits. And these varied across the gamut of things that people tend to measure. So we had, for instance, blood pressure, we had lung function tests, we had biochemical measures such as fasting plasma glucose, cholesterol, HDL, LDL, triglycerides, a variety of different factors like this. We measured the degree to which someone was apple-shaped or pear-shaped, the waist to hip ratio, and also a couple of other slightly different measures, educational attainment, so just how long did someone spend in full-time education, and indeed another measure called Spearman's G, so it's a measure of cognitive function based on a minimum of 3 different cognitive domains, and you simply take the first principal component of these test results and it gives you a generalized measure of cognitive ability. So we were able to recruit over 100 different cohort studies around the world. We divided them up according to continental ancestry or which particular genotyping platform they had used. And we were quite proud to have 350,000 research participants and 3 million data points across these individuals for these 16 traits. Here you can see now the forest plot for height. So that's all the different cohorts lined up there. And I think you can begin to see that there's a relative amount of consistency, and the summary point at the end is very, very significant. And we basically recapitulated our earlier work. This is work by colleagues of mine, particularly Peter Joshi, who's pictured there on the left. But we also found things that were much more interesting, I should say, than height. If we look here at this sort of multi-trait forest plot, the key point to think about is if the black box is near to, and if the little grey lines overlap with the 0 here, it means we see no effect. And given that we asked quite a few questions, I would say that all of these group here, we're seeing no effect. So that in itself is interesting. There was no effect of inbreeding on your blood pressure. There was no effect on your classical lipids, on your cholesterol. There was no effect on your insulin or on your glucose or any of these things. And we did, as I say, find the effect on height that we knew about, but we also found effect on forced expiratory volume in 1 second. So that's a measure of your lung function, of your puff, which had not been known about. But by far, the most interesting were these two here. Remember I told you about the cognitive function test and the education attainment. We saw very, very strong negative effects of runs of homozygosity on people's cognitive ability. And I have no doubt that the educational attainment is simply reflecting cognitive ability. These effects were reasonably small, but they still would be quite important if you were marrying your first cousin. We inferred that the offspring of first cousins would spend 10 months less in education, would be 1 centimeter shorter, would have 140 mils less lung volume, and would be 0.3 standard deviations. So that's about 5 IQ points lower were they not inbred. So these, while they are small, they're not entirely insignificant. And this paper received quite a lot of attention on Twitter. We had a small Twitter storm in Japan, actually, which I can't understand what the tweets were about, but I do know now that 5% of Japanese people, even in large urban areas like Tokyo, are the offspring of cousins. So I imagine this is why this interest arose. We now have a new iteration of the RhoGen consortium with a paper that's under review right at the minute. We wanted to expand out our census of traits, our survey of the potential effects of inbreeding. So we looked at 50 commonly measured traits that were either of medical interest or in some cases of evolutionary interest. And we looked at another 50 that were much more rarely measured, but they were available to us in the very large UK Biobank survey of 500,000 people. So we were able to put together a much, much larger data set, 1.4 million individuals. We distributed an automated software pipeline to all the different analysts all over the globe so that they could perform uniform and repeatable analyses in each local cohort. They then returned those files to us where we could do meta-analysis centrally. And I just wanted to show you down here a little bit more evidence of us all being inbred. The UK Biobank survey is a survey of 500,000 people aged between about 40 and 65, principally in England. So these are people who live in towns who are not particularly inbred, but the average person in UK Biobank nevertheless has 5 ROH. And here you see the distribution. So they've got an FROH of about 0.3 here, 0.3%. So that's equivalent to about third cousins once removed. I'm not for a second suggesting that everyone in England is each other's third cousin, but all of these kinships, very, very far back in time, are summing up together to add up to that sort of level. So which traits did we look at? I think a rather broad selection, trying to touch on many different organ systems. So we have anthropometric traits, we've got blood pressure traits, cognitive traits, we have traits of the blood, various traits to do with the electrocardiogram, traits to do with fertility, further cholesterol-based traits, some sort of behavioral traits, for instance, smoking and alcohol usage. We also have generalized sort of health traits, for instance, walking pace and so on, lung function traits, life history traits such as age at menarche, age at menopause, kidney function traits, liver function traits, traits to do with inflammation, traits to do with glycemia, and finally some traits to do with the function of your eyes. So a very, very broad survey. Obviously, the sample size varied by trait because not everyone measures everything in every study, and some studies didn't want to analyze all the different things that they had. So as you're getting used now, I hope, to looking at height, we first had a quick look at height to check everything was in order. It's almost a positive control. This is our new form of a forest plot, and we realized if we order the different cohorts by the power that they had, we get this lovely plot that Peter Vischer has christened a spruce plot, because it looks rather like a spruce tree. And basically this is showing that the new analysis has worked, and we have rediscovered the things we found in the last one. However, we also found really quite a lot more. Fully 25 of our 100 traits reached Bonferroni corrected significance. So we've actually found a much broader series of traits than I ever thought possible are actually influenced by homozygosity and therefore influenced by inbreeding. We can group them into a number of groups. We've got a series here which are related to really the size of the human body. We already knew about height, but actually weight is also influenced. So people who are the offspring of first cousins are about nearly a kilo lighter, all other things being equal, than their non-inbred kin. We see, again, the cognitive findings that we had, but this time we added reaction time in, which is a correlate of cognition, and the inbred individuals, the more inbred individuals, had slower reaction times. We see very strong effects in a series of traits to do with fertility. So the offspring of first cousins have about 0.1 fewer children. They have first sex for the first time 11 months later than their outbred kin. We also see some rather interesting traits to do with risk-taking. So things like speeding on the motorway or the number of alcohol units per week are decreased in the offspring of inbred individuals. And then there's finally a rather heterogeneous group of traits that are in some way related to health, where we also saw differences that were significant. Now, I think the strongest effect that we found was the effect on fertility. So we looked into this in a little bit more detail with some of these binary traits. For instance, whether someone had ever had any children or not. And in fact, we discovered that the effect of the runs of homozygosity on the number of children was almost entirely due to an effect on ever having children. So if you did have children, the number and the amount of homozygosity that you had did not determine in any way how many children you had. It just had an effect on whether you were able to have children. In fact, the offspring of first cousins were 55% more likely to be childless than fully outbred individuals. And it did not seem to be because of their ability to get married or their opportunity to have children, because we saw absolutely no effect of homozygosity on whether people were married or not. And we did see this effect on ever having children on people who were both married and unmarried. So it seems to be something to do with their ability to have children rather than their opportunity to do so. Of course, in this genetic work, as in many others, and I would say particularly so in this work, we need to be careful of the fact that other findings may be explaining our answers, whether there's any confounding in any way. And while it's not in observational sort of studies like this ever possible to be completely certain that you can exclude it, we did a number of stratified analyses to sort this out, and we were very happy to see that we had very similar effects when we divided these populations up by continent. We had very similar effects in very inbred populations, just as we did in very outbred populations. We saw similar effects, for instance, on height between people who were highly educated and people who were not so educated. We also became interested in whether it was the very long runs of homozygosity or just any kind of homozygosity that was having this effect. Traditionally, people would calculate inbreeding coefficients using a measure of heterozygosity SNP by SNP. So we actually calculated such a measure outside of the ROH. And we then divided the ROH up into those of quite short length, between about 1.5 and 5 million letters of DNA, and compared those to the very, very long runs. And what we see here is that all of the signal is coming from the runs of homozygosity, from the very long ones and also from the somewhat shorter ones. And there's hardly any signal coming from the homozygosity outside of these ROH. So while this is not definitive evidence, it does imply to me that it's because of very rare genetic variants that are being brought into a homozygous state by these runs of homozygosity. So it's probably not variants that we are genotyping in our data. It's because of variants that we have not observed because they're so rare, but they're nevertheless being brought into a homozygous situation by this run of homozygosity. So in summary, I hope I've convinced you that we are all related because of the ancestor paradox. I think it's obvious also that smaller populations share closer ancestry, and eventually anyone who is someone's ancestor is actually everyone's ancestor. So we're therefore all inbred, everyone in the world, even Barack Obama. It's just a matter of degree. And this inbreeding generates ROH in our genomes. They're ubiquitous. Everyone in the room will carry ROH. Populations and individuals in this room and in any other room probably vary rather dramatically in their burden, their individual burden of ROH. I don't think we have any Native Americans here, but they have the highest burden, and those of mixed race heritage have the lowest. This increased burden of ROH, it decreases bodily size, weight, height, and these things, and it also decreases cognitive function. It also influences a rather broad range of complex traits, particularly those related to fertility, but also, rather interestingly, those related to risk-taking. But if you think about it, having sex is a risky activity, and I wonder if these two things are actually reflections of the same underlying latent trait. The offspring of first cousins are 50% more likely to be childless than someone who is outbred. So that's really a quick run-through of our findings. All it remains for me to do is to acknowledge a variety of different people who are involved in this. Sequential PhD students of mine, Ruth McQuillan, Mirna Kirin, Peter Joshi, who is not there, and David Clark, who's presently a student of mine, standing here when we had a trip to Orkney. I also want to thank all my colleagues across the world, over 400 investigators who run cohorts like mine and who joined in this analysis, and of course the 1.4 million research volunteers who took part in these studies. If you're interested in updates on the sort of work that we carry out in Edinburgh in this group, you can follow us on Twitter @VikingGenes. We're launching a new recruitment drive, so quite a lot of this might be about recruiting new people, but we'll try to also update when we have papers published and so on and so forth. Thank you very much. I'd be happy to take any questions that anyone has.
Thank you, Jim. Very interesting. Questions? Frequency of chiasm or recombinations are will be different in different sites. Have you tried to analyze the size of region of homozygosity in these different chromosomal regions with high frequency of recombination and low frequency of recombination?
Yeah, we have performed a variety of regional analyses looking along chromosomes.
Yeah.
And we can see a variety of patterns that the amount of ROH decreases at the ends of the chromosomes.
Increased.
Going down, I think.
Decreased.
Yeah, decrease. We haven't finished these analyses. They became considerably more complex than we really realized. Initially, we were hoping to see if we could map where in the genome these effects were coming from. And in some cases, we have been able to do that, but we're still really working with this data. I don't have all the answers because we haven't finished doing this analysis. But there's certainly strong effects of recombination rate and a variety of other genomic parameters that are influencing the probability of ROH being in a specific place. So that's certainly true, and I think that's definitely influencing the distribution. But there's more going on than that. Remember in the early slide, I showed you the outbred people, and they all had an ROH in the same place. So we see that, and we call them ROH islands. I assume it's some sort of signal of natural selection. There's one at the lactase gene. There's a huge one at the HLA. But there's other ones in places that I've never heard. It's just some genes, some random genes. I don't know what's going on, but there's definitely a story there that needs to be investigated and written.
Yeah, thank you.
So it's I'm continuing the question. So it's not just regions of very high LD, you say.
No, but when we account for LD, we still LD influences it, sure, but we still see it. Yeah.
You see it in lactose gene.
Yeah.
Wow. Interesting. More questions?
How it possible to explain that Drosophila strains can live in conditions of inbreeding many thousands generations without influence on the physiological traits?
Yeah, I mean, there are many theories about this. I guess I'm as surprised as anyone that some of these people are walking around with so much homozygosity. In UK Biobank, we found someone 33% of their genome is homozygous. And this person is a healthy volunteer who gave consent, who went to a clinic. It would appear that inbreeding isn't as bad for you as people think. And you know it's a roll of the dice. If you don't happen to have any recessive lethals or nasty variants, it doesn't matter if your DNA is homozygous. Some people believe in purging, whereby they can purge out these deleterious alleles. I have yet to see evidence of that particularly. But I think inbreeding isn't as bad for people as is generally considered to be the case. So, I mean, it's very hard because you're rolling a dice. If you happen to have a cystic fibrosis variant, it can be a disaster, obviously. But if you don't, maybe you can get away with it. I don't know if that was the answer to your question.
Other questions?
Really, it was very interesting. But maybe there is some difference if there is some population, small population, for example, in Iceland, and they are living a lot of time together and high level of inbreeding, and it's necessary to imagine some selection. Yeah, and maybe it will be a little different homozygosity, then it will be formed just random. When we have some cases with hybridization of disrelated people. Maybe it will be a little different type of homozygosity.
Yeah, I mean, these things are certainly possible. It relates to your earlier point about where in the genome, because this standardized form that we think about here would be very random. But if something unusual has gone in an island population, we would maybe see a signal of that piling up in one part of the genome, perhaps. We have yet to look.
Yeah, thank you for the lecture. I have a question concerning this part about cognitive functions and education. Can it be not the matter of genetics, but to deal with some social opportunities of these people who live on some distant islands and tribal culture or something like that?
Yeah, of course we have to be very careful of confounding in that way, but we didn't actually ever compare people with distant islands, on distant islands, to people in the cities. The comparison was done within the distant island, and the comparison was done within the city, and they were then meta-analyzed, so we are protecting ourselves in some way. We also actually did another analysis that, I don't know, the slide didn't make it in here, which finally has nailed these sorts of things. Because those of you who are geneticists will know that siblings vary in the amount of homozygosity, in their realized homozygosity. So we did analyses within sibling pairs, and we're able to show for the strongest traits, for height, and also for educational attainment, that there was a very strong association with homozygosity, and we've controlled for all potential confounders except things that vary between siblings, which you're getting into special pleading then, to be honest. So you're right. There's a lot of room for confounding, but I think we finally nailed it with the within sibling family experiment. Obviously, the power is really, really low. A random pair of siblings differ by 10 megabases in their homozygosity. So you need thousands and thousands and thousands of sibling pairs. And so it was only for the effects that were strongest have we been able to see it. If anyone has 100,000 sibling pairs genotyped, I'd love to hear about it, because we could finally nail all of this. But even with our 1.4 million individuals, we were just at the edges of power. But basically 75% of the effect was genetic, and 25% of the effect was probably due to some form of social confounding. So you're right, but I'm also right.
So it was a great question, but also this sibling thing is a very strong argument.
If we consider supergroups, human supergroups, as I take it from the literature, from the papers already published on 1,000 genomes, I can see that East Asian population have small, relatively small variation in terms of heterozygosity. Could you share some words on that?
Certainly, I would tend to agree with you. Of the very major continental population groups, that East Asians are perhaps the least diverse. But there are many other groups in the world that are lower diversity, but they're probably not occupying an entire continental landmass in this way. But in terms of homozygosity, they're not hugely different from Europeans. I mean, Europeans are not exactly genetically diverse. If I take 100 Africans from any village, they will have more diversity than probably the people in this room. So I don't know that it's East Asia that's particularly different. It's Africa that's so different from everywhere else. Europeans and East Asians, yeah, we're not particularly diverse, I wouldn't say. We're not hugely different from one another either.
Thank you very much. And another question is about the chromosome Y squeeze in terms of 5 million years ago. There was a paper in Science, I believe, or Nature. Is there any interpretation? I'm sorry, I'm not directly addressing your topic, but
it's quite okay. I'm actually very interested in the Y chromosome. It's a very interesting and understudied piece of DNA, despite half the people in the room carrying it, and 35 million letters of DNA that no one ever looks at. But it's a very unusual and slightly difficult and irritating piece of DNA to work with. It has profound drift effects on it, such that FSTs are through the roof. The differentiation between populations is enormous. You can look very, very finely and tell people apart from a few miles on the basis of their Y chromosomes. There's one new marker on the Y chromosome every 3 generations. So in theory, you could build a genealogy of every man on Earth with a 3-generation resolution if you were able to sequence all of their Y chromosomes. So there's a lot to be learned actually about microgeography, about history, about this. And although the Y chromosome is somewhat old hat, I think its day actually has yet to come in terms of exploring these things. You'd be able to see very beautifully this history of the change in population size of men, but men need women to reproduce with, so it's still telling you something from the branching structure of this tree and where there are places where there's polyfurcations versus places where it's thinning down. I think there's a lot to learn there. I run another consortium called RhoGen, which exists to try to see if the Y chromosome influences any of my favorite 16 traits, but it's been rather disappointing because we see very little. Well, we found an association with height. It's always height for some reason in Japan. So there's a Y chromosome haplotype that's found only in Tibet and in Japan, and we see a very strong signal of that influenced height amongst the Japanese. We don't have any Tibetan cohorts, unfortunately.
More questions?
You said a lot about mixing of the people from different places, yeah? But there is some direction of migration. For example, there are a lot of migration from Africa and Europe, yeah? But from my point of view, there are only much less migration otherwise from Europe to Africa. Maybe it will be some special situation in the result of such directed migration. What do you think about this?
There might be. There's actually rather good genetic evidence for a back migration to the Chad Basin in the Sahel belt of northern Africa, where people came from the Middle East back to Africa about 7,000 years ago. But I don't have any DNA from these people. I just read about it in papers.
No, I mean not just for the past, but just for nowadays.
You mean the movement of people now? Yeah. Well, that's a challenge to genetics, isn't it? If you're interested in population history, you need to study it now, because by my grandchildren's generation, it's going to be completely mixed up. And it means that the homozygosity is going to be decreasing, and so people are going to get a centimetre taller, or a little bit taller, and a little bit cleverer. And
but maybe you can do some forecast for the future.
Maybe.
Okay, any more questions? Well, if not, I would like to thank Jim very much for this very interesting talk, and thank you all to be here and asking great questions.
Thank you.